All right, here we are back in the lab. Come into the lab. I'll wash my hands, put on my lab coat, sterilize my surface, into the biohazardous waste. Put my gloves on. All right, so what I will be demonstrating in this video is making a negative stain from our previous dish. And I'm not sure if the previous one I actually showed the test tubes in the test tube rack. All right, so my surface is clean. I have my slides. It's labeled. I'm properly holding the slide. All right, so I will begin with placing a drop of nigrosin. Oh, more than a drop. On the end of the slide. All right, then I'll take my inoculating loop, which has been sterilized for the 10 count, and then hold for 3. Ooh, tricky. Okay. On an angle, I gently am gathering the microbe, the specimen. And I place the loop gently in the dye, the nigrosin, and I just shift that loop laterally back and forth. All right. I sterilize my loop once more for 10 count before placing it down. All right, now I grab the other slide and I hold at a 45-degree angle and I bring it to the dye until it touches the dye, and then I drag it back carefully across the slide like so. All right, so when that drop flows across the width of the slide, I push gently and evenly. All right, so now this slide goes into the sharps container bin. And then this slide, we will place atop the steam bath until it dries. All right, clean up, same thing, remove the gloves, sterilize the surface first, sterilize the loop, remove the gloves, and take the coat off.